Hello, ladies and gentlemen. Welcome to the fifth session of How I Treat Autoinflammatory Disorders course. I am Dr. Kosa Asmashari, Assistant Professor of Pediatrics at Children's Medical Center, Tehran University of Medical Sciences, Iran, and I'm the head of the executive team for the course. This course is programmed as a progression to How I Treat series, an educational program planned by the leading house for Iran North America academic partnerships of TUMS. The program focused on a single disease or on one or more distinct aspects of a single disorder, and they offer diagnostic and therapeutic advice by enrollment of international, well-known experts in the field. Our course, How I Treat Autoinflammatory Disorders, is comprised of 8 to 10 sessions held weekly or bi-weekly, and the program will focus on various aspects of autoinflammation, pathogenesis, diagnosis, treatment, patient education, and network development. We intend to provide physicians, pediatricians, immunologists, and rheumatologists with an opportunity to access the latest updates on autoinflammatory disorders. We have gathered elite experts from around the globe, USA, Germany, Italy, Turkey, and Iran, to share their knowledge and clinical experience in each session. We hope these endeavors can lead to a better understanding of these newly introduced diseases, which are probably not as rare as they appear. I need to take a moment here and thank Dr. Vahid Ziai, head of our scientific committee, professor of pediatric rheumatology at Children's Medical Center, Tehran University of Medical Sciences, and the president of Pediatric Rheumatology Society of Iran for his full support of the course. We are also very grateful to Dr. Marjan Yaghmai, head of leading house for Iran North America academic partnerships, for her kind support. I would also like to express my gratitude towards Professor Someira Farman Raja, Professor of Rheumatology and convener of APLAR Pediatric Rheumatology Special Interest Group. Thanks to her support and cooperation, our course is now endorsed by APLAR. Asia Pacific League of Associations for Rheumatology, or APLAR, was established in 1963 in Sydney. The world-known association of APLAR strives to propagate and consolidate rheumatology endeavors in the Asia Pacific region. APLAR's mission and goals focus on providing the state-of-the-art care to patients with arthritis and other musculoskeletal diseases through the continuing professional development of members, increasing the awareness and understanding about rheumatic diseases, patient advocacy and empowerment, and fostering research in the field of rheumatic diseases. In order to receive a certificate for each session, you need to register via Appraise to Raise website. The link can be found here in the chat forum, in my slide, and in the session's poster. You can also register for the whole course and receive a certificate of attendance in an international autoinflammatory course approved and validated by Asia Pacific League of Associations for Rheumatology, or APLAR, Pediatric Rheumatology Society of Iran, Iranian Rheumatology Association, and Tehran University of Medical Sciences. For those who were not able to attend our previous sessions, there is a possibility to view the video of the session on the website, answer to the post test, and be eligible to receive the course certificate. We decided to provide course certificates to all the ones who either participate in the session or watch the video and pass the post tests, as the goal of our course is to educate the participants. Today's session is entitled The Role of Type 1 Interferons as a Trigger of Cytokine Storm in MAS. We are honored to have one of the most outstanding experts in the field from Germany, Dr. Christoph Kessel. As Assistant Professor in Immunology, Dr. Christoph Kessel is currently leading the Unit for Translational Inflammation Research within the Department of Pediatric Rheumatology and Immunology at the University Children's Hospital in Münster, Germany. Dr. Kessel graduated in biosciences and obtained a PhD from Goethe University, Frankfurt, Germany, on mapping of conformational B-cell epitopes and peptide-derived B-cell targeting approaches. In 2009, he joined Professor Rickard Holmdahl and medical inflammation research at Stockholm, Sweden, for postdoctoral training on the role of post-translational modifications in generating B and T cellular neoepitopes, driving break of tolerance in autoimmune conditions. Following his interest in studying particularly innate immune functions in inflammation in highly translational research settings. Sorry, I have a problem here. Oh, okay. He joined Professor Dirk Voll's team at Münster University Medical Center in 2012. Dr. Kessel is currently steering the SystemicJIA and macrophage activation syndrome working party of the Pediatric Rheumatology European Society, or PRESS, where he is responsible for coordinating science and research. The moderator of this session is Dr. Rahela Assari from Iran. Dr. Rahela Assari is an assistant professor of pediatric rheumatology in Children's Medical Center, Tehran University of Medical Sciences. Dr. Assari is interested in researches in the area of connective tissue diseases, diagnosis and treatment of autoinflammatory disorders, macrophage activation syndrome, and the recently introduced multisystem inflammatory syndrome in children. Dr. Assari's goal in research and practice is increasing awareness of parents and patients with connective tissue diseases and ameliorating the outcome of such disorders. She is a member of Pediatric Rheumatology International Trial Organization, or PRINTO. I want to ask you, our dear participants, to share your ideas and ask your questions in the chat board. It's better if you address your questions to Dr. Assari, the moderator. We will now begin today's session. Dr. Assari, we are very eager to hear you start the session. Thank you very much.
Hello, dear colleagues. Thank you, Dr. Kosa, for introducing me. First of all, I would like to express my gratitude to supporting group APLAR, especially Dr. Somera, Tehran University of Medical Science and Pediatric Rheumatology Society, and the head of this, Dr. Zia. Yes, our session are entitled How I Treat Autoinflammatory Disorder. We know that autoinflammatory disorders are new sciences in recent years and are developing day by day and year by year in pathophysiology of this. And we know that they are the hereditary disorder. Sometimes we have some problem in treatment of this. And as clinician, when we know the pathophysiology in details, it would guide us and give us the clue to the treatment of these disorders. Macrophages activating syndrome, we know that is a crucial situation in the background disorder, autoimmune and autoinflammatory disorder. And as early as possible, we detect and diagnose this. It can help us rescue and save our patient. Yes, we know that interferon gamma has a critical role. Before this session has the critical role and initiator of waterfall of cytokine storm from CD8 plus to stimulate the macrophages for cytokine storms. And we are honored to have Dr. Kessel. But what is really the location of interferon 1 in this situation? We reviewed the manuscript of Dr. Kessel. I really enjoy of them, and I really suggest it to my dear colleagues. I like your new phrases, innately adaptive or truly autoimmune in SystemicJIA. I really like this. About in SystemicJIA that describe the contribution and correlation of adaptive immune system and innate immune system. Firstly, we have systemic manifestation, innate immune system, and it's truly progress to adaptive immune system involved with the helper 17 and the others. Yes, what is really the location of interferon 1 in macrophage activating syndrome? We are eagerly to listen. Let's listen together. Thank you very much. I hope you'll enjoy this.
Thank you very much for this extremely kind introduction, really, and Dr. Kosa. I'm really honored to be with you today. Thanks a lot for reaching out, and thanks for the faculty of your course for supporting this and for having me. And now I will share my screen, and we already just tried that and it did not work. Let's hope it works in the way that I want it to work right now. All right. Toggle it a little bit. So, Taiwan interference in macrophage activation syndrome. That's what we want to talk about today. And looking at the program of your course so far, you have touched upon some autoinflammatory diseases very specifically. Last lecture, last time you had an outstanding lecture by Eike from Bonn in Germany, which was not that disease focused, but he provided you with an overall overview on inflammatory mechanisms and inflammasome function and how that integrates, of course, in autoinflammatory diseases and is influenced by many factors, including lifestyle. Today, what we're going to do is we will look at a disease condition again more specifically and combine that with some mechanistic or the mechanistic knowledge which we think we have today. So I think it's probably a really translational lecture in course of your whole course program. And I divided this lecture into 3 chapters. So first we will look at macrophage activation syndrome and the family of cytokine storm syndrome, sort of to put us all on the same level of pathophysiological understanding and the point where our questions originated from. Then we move on towards interleukin 18, IL18, which is very central to MAS. Pathophysiology, Dr. Reedy already mentioned interferon gamma. We for sure also touch on interferon gamma, but this lecture will be a lot on IL18, so we will have to look at this a bit more specifically in chapter 2. And chapter number 3 will then get us to what we really want to talk about, the role of Taiwan interference in cytokine storm scenarios and macrophage activation syndrome specifically. And in fact, the topic of this session couldn't be more fitting for us in Germany right now, because we're experiencing severe storm conditions as we speak. There is a major warning by the German weather authority that in the northern part of Germany, where we're sitting right now, we're going to have extremely heavy winds coming in over the night. We already had that yesterday. The media is freaking out. Schools are closed. So it's, as I said, it couldn't be more fitting. Beyond that, I think nothing, or there has been very little in the past 2 years that has received more general attention, and at the same time also created as much awareness in the medical society globally, but also in society as such, as the term cytokine storm syndrome, as well as macrophage activation syndrome, pretty much or almost exactly 2 years ago. It was a commentary in Lancet that alerted the global medical community of the fact that in course of SARS-CoV-2 infection and COVID-19, it may be, in fact, cytokine storm conditions that drive this severe condition. And ever since that, this has, as you all know, of course, has been a very central aspect in understanding COVID's pathophysiology. Spoiler here, this is the last time that I'm going to talk about COVID in course of this lecture. We can discuss that in course of the discussion if you want. Now, in the very conditions we want to discuss in course of your course, and we're actually mainly working with inflammatory conditions. I think it's this paper which came out in 1970 by Schaller et al. That first described a condition in actually 5 children, a inflammatory condition involving the liver in children with rheumatic diseases. And these conditions occurred with disease onset and were associated with high fever, arthritis, a rash, splenomegaly, and lymphadenopathy. And then this was followed by 2 case reports in Annals of Rheumatic Diseases, now already linking this to Still's disease, that described a very similar phenomenon. So here it's a 12-year-old girl with a hemophagocytic syndrome, and in this case, it's a 15-year-old boy who was described as gravely ill due to erythrocyte phagocytosis, and this was already suspected back then as virus associated. Now, today we know that what these reports were talking about is macrophage activation syndrome, or we call it macrophage activation syndrome, and it's a vital part, as you all know, of the inflammatory continuum in Still's disease, where we have like a more febrile condition, systemic condition, primarily driven by innate immune mechanisms, and eventually this can progress towards a chronic destructive arthritis. Arthritis can already be evident at the beginning. We know that, the clinical picture can be very diverse, and currently, the view is, and that was what Dr. Rahidi was actually pointing at in your introduction, currently the view is that this more arthritic type of Still's is driven by adaptive immune mechanisms, or as we like to term them, innately adaptive immune mechanisms, which is mainly T cells that we cannot really put into like the innate immunity or adaptive immunity drawer. They're sitting kind of somewhere in between. And that's why we phrased it innately adaptive. But it's then T cells involved in this conversion of the disease eventually. And on top of that, we basically always have a threat of patients to fall into macrophage activation syndrome, which is combinedly driven by innate and adaptive immune mechanisms in the end. Now, MAS is most prevalent, certainly in context of systemic JIA. It's about fulminant mass in about 10% of cases. 30% of systemic JIA patients develop subclinical mass, which may not result in the patient to end up in the ICU, but it can still cause severe clinical problems in managing those particular patients. And apart from systemic JIA, mass can also be seen in Kawasaki syndrome, which, by the way, shares kind of a lot of pathophysiological traits with systemic JIA, and it's in part even misdiagnosed one for the other. And we also have mass in SLE. Also, that is certainly less frequent. In systemic JIA, the overall mortality, particularly with the fulminant case, is about 8%. And you know that according to the latest ACR-EULAR criteria, mass in context of SJA is diagnosed by, of course, the presence of persisting fever plus extremely elevated levels of blood ferritin, and combined with a drop in overall thrombocyte counts. And from a pathophysiological point of view, we are dealing with an overactivation of macrophages that then start to phagocytose erythrocytes and neutrophils and platelets, so reduce a severe cytopenia all over the board, basically. And this overactivation of macrophages is triggering a cytokine storm. I just love this picture of a cytokine storm, because I think it's really to the point what we're talking about. Cytokine storm syndromes is actually an umbrella term. In fact, and here I'm referencing the cytokine storm umbrella that has been drawn by Scott Canna and Lauren Anderson when they discussed that in COVID context in early 2020. So under this umbrella, we have macrophage activation syndrome as a condition we see in context with rheumatology. We have primary hemophagocytic lymphohistiocytosis, which is a condition that is normally seen with oncologists. Here we are dealing with mutations in genes that encode for enzymes in the lytic mechanisms of NK cells, so that help NK cells to lyse target cells. And the NK cells in this case are dysfunctional, which leads to an overall overactivation of macrophages. These patients are usually treated and seen in oncology, and lately we also have CAR T-cell therapies that can result in cytokine storm conditions, also seen with oncologists. And then we have a number of different conditions that usually associate with any kind of infection, which can be virus, bacterial infections, or other malignancies that are actually termed secondary hemophagocytic lymphohistiocytosis or infection-induced HLH. So all these conditions group under this umbrella, and it's the differences in nomenclature for conditions that are highly overlapping can be very problematic in times. Because in fact, all these conditions share a large part in their immune, in their pathology and in the immunological presentation. Because regardless of the genetic susceptibility, which can be single genes, as highlighted in terms of primary HLH, or completely undefined sets of genes, as in SystemicJIA, acquired susceptibility, which can be on-top infections, as well as triggers, it all ends up on a very central and overly prevalent immunological axis, which involves macrophages, it involves CD8 positive T cells and NK cells, and that together are being overactivated by cytokines, which are interferon gamma and IL18. And these overactivation then gives rise to a large overlap in the clinical presentation of these syndromes, which impairs or can impair an early on and precise diagnosis, because the clinical presentation is so similar. So IL18 plays a central role in the immunology of these cytokine storm syndromes. So for a moment, we should step back and have a closer look at IL18. Again, just to level us in knowledge. So IL18 is a member of the IL1 family of cytokines. It is a bit smaller than IL1 beta itself. It has a pro-inflammatory function, and in terms of structure, the overall folding of IL1 and IL18 is actually quite similar as well. You still record from the lecture of Eike Lutz on Friday 2 weeks ago, that IL1 family cytokines, IL1 and IL18, and the release and the production of those is involving inflammasomes. So here is an example of the classical NAP3 inflammasome. So we have a stimulus which can be damped, so a damage-associated molecular pattern arising from cells being damaged and releasing their content, or a PAMP, which is a pathogen-associated molecular pattern, which can be LPS, for example, so any bacterial or viral structure. And these signals come in via pattern recognition receptors, which can be a TLR4 here in case of LPS, for instance, and then activate transcription factors that produce inflammasome components as well as the pre-products for inflammatory cytokines such as IL-1 beta. Then we have inflammasome assembly in case of NAP3 upon a second signal, a stress signal to the cell, and this inflammasome generates a mature caspase-1. And caspase-1 then cleaves both pro-IL1 and pro-IL18 peptides into mature IL1 and IL18, which are then released, as we know since the discovery of the Gauss-Dermans, where porous in the membrane. So the caspase-1 not only cleaves the propeptides of IL1 and IL18, but also cleaves Gauss-Dermin D and processes this so it can assemble the pores in cell membranes to release cellular content, including IL1 and IL18, presumably by a simple size excluding mechanism. Now, once IL18 has been released from cells, it signals actually predominantly to T cells. We also have, this is IL18 receptor expression according to data from the Human Protein Atlas. We also have IL18 receptor on granulocytes as well as epithelial cells, where it probably ensures membrane integrity. But most knowledge with respect to the downstream function of IL18 has actually been collected with respect to signaling to T cells. IL18 was formerly known as interferon gamma inducing factor, and this is actually still its widely reported main function. So T cells that are exposed to IL18 are stabilized in their interferon gamma expression, and it increases interferon gamma expression. It's also reported to increase IL-17 release from gamma delta T cells. So from our data, we can say that it doesn't necessarily push the IL-17 release and production by these cells, but what it does instead is, and that's something that IL-18 also does to other T cells, it helps T cells to proliferate. So this is a plot that illustrates proliferation of gamma T cells when exposed to different cytokine stimuli. And the higher the peaks are getting here, the more the cells are proliferating, though they start proliferating here. And the higher the peaks are getting over here, the more cell proliferation you have. And if there is no stimulus, you basically have very little proliferation. It's a little bit enhanced with IL-1, but as soon as IL-18 comes in, you have substantial gamma delta T cell proliferation in this respect. And this is also true, also shown widely by other people for other kinds of T cells. So it's IL-18 has a central role in maintaining T cell function and facilitating T cellular interferon gamma expression. It binds the IL-18 receptor in a very similar manner as IL-1 binds the IL-1 receptor. And I like these drawings and visualizations of the receptor because in many occasions, the receptor is drawn in a way that you think IL-18 comes from the top. And in fact, both IL-1 and IL-18 come in from the side to the receptor and bind like in a binding pocket to both the IL-1 R1 and the IL-18 receptor respectively. And I think that IL-18 signaling can also be opposed in a very similar fashion as that is true for IL-1. So for IL-1 signaling, there are anti-inflammatory mechanisms that can help to cool down inflammation. So there is the IL-1 receptor type 2, the IL-1R2 that functions as a decoy receptor to circulating IL-1 beta. You deplete IL-1 beta very efficiently from the circulation. And there is, of course, the IL-1 receptor antagonist, IL-1RA. We all know the drug that is coming from this, Anakinra, that is sitting in the IL-1 receptor and blocking the access of both IL-1 alpha and IL-1 beta to the receptor and preventing signaling that way. And for the IL-18 signaling pathway, there is a similar molecule around like IL-1RA. It's called IL-18 binding protein and operates in a very similar fashion. So it actually captures circulating IL-18 and depletes IL-18. It's a very tight binding of IL-18 binding protein to free IL-18 and forms a very stable complex, and thus reduces the overall bioactivity of this cytokine. Now, in context of systemic JIA, we can discuss IL-18 actually from two angles or two aspects or important about this cytokine. So we can discuss it from a biomarker point of view, and we can discuss it from a pathomechanistic point of view, which are linked, of course. We'll get to that. Now, the first biomarker data that closely associated IL-18 overexpression in systemic JIA or associated systemic JIA with IL-18 overexpression have been generated by our colleagues in Utrecht in the Netherlands that reported back in 2009 that in systemic JIA there is a profound overexpression of IL-18, and that is also very specific for this JIA subtype. It cannot be seen with oli or polyarticular JIA, where other cytokines are elevated, but not IL-18. IL-18 overexpression is very systemic, very specific in systemic JIA. And meanwhile, this has been reproduced very frequently, and this is just another piece of data that have been generated by Grant Schulert and Shima Yazin in Cincinnati and reported in Rheumatology back in 2020 that also show that there is elevated IL-18 in systemic JIA, and it can separate active disease from inactive systemic JIA with a very good area under the curve. And it can even tell or report patients that had a previous history of MAS versus patients that have never experienced macrophage activation syndrome. Now, we have also contributed in this respect in a recent study where we addressed the question whether within the group of hyperferritinemic cytokine storm syndromes that have this very profound immunophenotypic overlap, we could still tell apart systemic JIA from hemophagocytic lymphohistiocytosis, both primary and secondary, which is important when, for instance, you see as clinician, you see a patient for the very first time, and he or she's coming in severe, in bad condition with obvious cytokine storm features, and you want to know, is this secondary HLH or is it SJA associated MAS, because that can then substantially influence your further treatment decisions, of course. And in this study, we looked at a lot of different biomarkers together with our colleagues from Utrecht again. And we could separate signatures on the level of this blood biomarkers that can be associated with secondary or primary HLH, as well as systemic JIA. But this signature was also seen with healthy controls. And there was one additional signature that was then really specific for systemic JIA, and kind of reflecting the overall immunophenotypic overlap I already touched upon now several times, was kind of the picture of these cytokine signatures, because even though you can like delineate your different blood biomarker signatures, you still have quite some overlap between MAS and these different HLH conditions. Nevertheless, on the level of single markers rather than signatures, after an extensive validation process, we identified IL18 plus some other factors or combinations of IL18 with CXCL9 and CXCL10 as powerful discriminators between systemic JIA mass and primary as well as secondary HLH, and particularly with respect to separation of mass from infection-induced HLH. These markers like IL18 and S100A12, which is a granulocytic protein, could separate mass from secondary HLH more powerful than the already established MH score, which is a clinical score for diagnosing mass and HLH, or can support the diagnosis of mass and HLH. And you can see, compared to this red line here, that this is works very efficiently with these blood biomarkers. And with respect to IL18 and the ratio with IL18 to CXCL9, we're in fact reproducing data that have already been reported by the group of Scott Kanna back in 2018 in blood, because they already described that the very same thing, in fact, that there is a very powerful separation of systemic JIA-associated mass from primary HLH with using these parameters, as well as separations not as good, but there's also a separation from infection-induced or infection-associated secondary HLH. So MAS or IL18 is a very potent biomarker for assessing MAS activity, as well as for supporting its differential diagnosis. And the reason for this strong prevalence of biomarker role and power of IL18 for MAS pathophysiology is actually rooted in pathophysiology, because if we look at this inflammatory continuum in Still's disease, and we overlay the currently known pathophysiological traits in here, we can see that IL18 has a very strong and prevalent role in disease pathophysiology. So the overactivation, the inflammatory overactivation of innate immune cells results in massive IL18 expression, which is then driving its downstream function. So biomarker role and biomarker impact of IL18 is, of course, directly linked to its fundamental importance in disease pathophysiology. And that's what we call a strong mechanistic biomarker. Now, IL18 is, or the pathophysiological role of IL18 is that it is a central driver of the cytokine storm in mass. So it's originating from myeloid cells together with other inflammatory cytokines. And then wire the IL18 receptor expression, which is so prevalent on NK cells and other T cells, induces interferon gamma, which then together with GM-CSF fires up more activation of myeloid cells. And we have this whole vicious circle of rolling in this cytokine storm. And the main knowledge on the central role of IL18 in MAS pathophysiology has been generated by these two guys, in fact, which is Scott Kanna now in Philadelphia and Cem Gabay in Switzerland, who described in two back-to-back publications of blood in 2018 that There is, of course, inflammasome activity and expression in myeloid cells, but also in intestinal epithelial cells. And that results in expression of IL-18. And in the end, it's this free IL-18 that signals. So this is IL-18 that is not complex by IL-18 binding protein. It's free IL-18 that is required to fire up the cytokine storm in MAS, because only in its free bioactive form it can engage with the IL-18 receptor on NK cells and T cells and induce interferon gamma expression. And that is a condition or a scenario that is particularly the case in NLRC4 mass. So this is a very special form of macrophage activation syndrome associated with monogenic genetic defects in NLRC4. This is the case in systemic JIA as well as in adult onset Still's disease. And it's something we do not see in other monogenic autoinflammatory diseases, such as FMF, for instance, where we also have very high IL-18 levels, but it's usually complexed and blocked from its signaling to T cells by IL-18 binding protein. And this is also not the case in primary HLH, where we also have elevated IL-18 levels, but again, IL-18 is sequestered by IL-18 binding protein because they are more or less in balance in this scenario. And the power of capturing free IL-18 in preventing inflammation has been demonstrated by Scott and Behrens and Cem Gabay again and other colleagues in a case with NLRC4 mass, and that patient has been treated at some point with recombinant IL-18 binding protein. And you can see here on the level of CRP and ferritin that after initiation of this treatment, we have a reduction in CRP and overall ferritin levels, while at the same time, the free IL-18 levels are brought down once the IL-18 binding protein comes in, and the overall CXCL9 levels, which serve as a biomarker for interferon gamma signaling, are also being reduced in this by this therapy, and the patient overall improved in inflammatory status. Now, the collective evidence, in fact, for a really dominant role of this IL-18 interferon gamma axis in systemic JIA math pathophysiology are overly prevalent, I think. So we have the biomarker data already touched upon, and we have the studies, even more important at this point, I think, that interfere with either IL-18 signaling, or I didn't touch upon this specifically, do that now, or interferon gamma signaling. So this is preclinical studies as well as the trials for involving imapalumab, so an antibody that targets interferon gamma, ongoing, but the data already imply that it's a very powerful drug in controlling hyperinflammation. So we have all this in place. What has been or remained a black box is the regulation of IL-18 expression, and here I'm talking about gene expression rather than the processing, the protein processing I already touched upon. So how is IL-18 expression in cells being controlled has been fairly unknown and still not fully solved. And in this respect, we undertook a study that is actually quite a roller coaster ride, and I'm trying to guide you through it because it emerged actually from observations we made in sepsis, and then eventually evolves towards macrophage activation syndrome. So the accompanying editorial to this paper actually highlighted that we're touching upon extremes of inflammation here to understand the overall transcriptional control of this cytokine, and also what that might mean in terms of therapeutic relevance in the end. So the first observations we made, or the initial observations we made, were done in a unique study that has been run by Peter Pickers in Nijmegen in the Netherlands, who immunized or injected LPS into healthy human volunteers. It's so-called in vivo endotoxin re-challenge model. He applied that previously to study treatment in sepsis. These patients are constantly monitored in the clinic, develop flu-like symptoms, and we had the opportunity to look into cytokine expression in the plasma of these patients. And what we could see is that upon initial LPS injection, you have an elevation of inflammatory cytokines, and upon LPS re-challenge, we don't see that increase anymore. So this is something that is classically termed as endotoxin tolerance, or endotoxin-induced immune paralysis. So it's a textbook reproduction, basically, of already known findings. What we observed in addition to this was on the level of IL-18, that IL-18 seemed to be untouched by this immune paralysis. It even seemed to benefit from this additional exposure to LPS, and that was something that surprised us. And we rebuilt this experiment in the lab using cells, monocytes here from healthy donors, and stimulated those monocytes repetitively with LPS. And we could see the exact same thing now on gene expression level, as we could see in the plasma of these in vivo endotoxin re-challenge patients, which is that upon LPS re-challenge, we see the immune paralysis on the level of TNF and IL-6 and IL-1, but we don't see that on the level of IL-18. In fact, and now here it's really prevalent, the double hit by LPS was really pushing up IL-18 in a way as we have not seen it before. And we thought that was really striking, because I said, and IL-1 and IL-18 are members of the same cytokine family. They undergo the same mode of processing and secretion, and they're actually co-expressed in many autoinflammatory diseases. So when people talk about elevated IL-1 levels, it usually implies it's also elevated in IL-18. So they are kind of like identical twins in this disease, regardless of the underlying mechanism of the autoinflammatory disease you look at. These cytokines are usually co-expressed. So we thought it's really striking that we see this obviously really stark difference in their mode of expression. And we followed up on this. And what we could see in the end is that the reason for this difference in expression and upon LPS re-challenge was actually a striking difference in gene expression kinetics. So with IL-1, we have like a really early expression of the IL-1 gene, transcription of the IL-1 gene upon LPS challenge in human monocytes. It's like, it's coming ad hoc, basically. But for IL-18, we have a strikingly attenuated, like delayed kinetics, which is not happening in any way as the IL-1 transcription does. And we thought, well, this is something we need to understand further. So the first thing we looked at was whether it may be influenced, IL-18 expression may be influenced by other cytokines that are coming even earlier, maybe upon LPS challenge, like TNF. Is TNF involved in driving IL-18 expression? Questions like this were addressed by us. And what we could see in this series of experiments, that none of the cytokine blocking drugs had any effect on lowering IL-18 expression. The one drug that was working powerfully in this set of experiments was tofacitinib. So tofacitinib was the drug that could actually bring down IL-18 expression at the same time what was actually elevating IL-1 transcription. So you might ask here, so why is tofacitinib working in a scenario where you stimulate with LPS? And then tofacitinib is a drug that is blocking JAK-STAT signaling, which is coming in mostly via interferons. And the reason why it's operating in an LPS stimulation context is this: LPS, usually when it hits a cell, the signal goes in via TLR4, activates NF-kappa B, inflammatory cytokine expression is the result of this. But what is already happening within minutes is that the TLR4 receptor is actually taken into the cell. And when this happens, a different pathway is being switched on, which is the TRAM-TRIF pathway. And this pathway is triggering interferon beta expression. So TLR4 signaling using LPS, via LPS, stimulated by LPS, is in fact generating interferon, type 1 interferons. And this is now exactly sort of the angle of attack for tofacitinib. So when these interferons upon this TLR4 internalization are being released, they can signal, of course, in an autocrine fashion to the same cell, but also in a paracrine fashion to another cell. And tofacitinib is interfering with this signaling of the type 1 interferons that are induced by LPS signaling here. So we had a good hint that type 1 interferons may be critically involved in IL-18 transcription. We followed up on this by taking human monocytes again, but priming them with type 1 interferons prior to LPS stimulation. And when we look at IL-1 in this set of experiments, we can see that there's basically no change in the overall transcription kinetics over time. They all look the same, regardless if the cells get to see interferon alpha or beta 4 hours or 16 hours before being hit by LPS. This is quite different with IL-18. Here you can see that the interferon priming is actually modulating the overall gene expression kinetics in the way that it moves it away from this the delayed expression to an early expression. So it's kind of pre-sensitizes the cells to make IL-18. And that does not work, I have to mention that, with interferon gamma. So it's a type 1 interferon specific effect. And in the course of performing these experiments, we also had the opportunity to look into a patient with a step 1 gain of function variant. And we also looked at IL-1 and IL-18 expression in this patient. And you can see that for IL-1, the IL-1 expression in this patient, as well as healthy controls, is basically unchanged. But for IL-18, we also see like a shift towards an early expression in this step 1 gain of function. So type 1 interferon, that was the reasoning at that point. Type 1 interferon signaling, number 1, and STAT activity, STAT1 activity, are crucial to IL-18 gene expression in human monocytes. And the final experience from this that I'm going to show you from this set is neutralization of interferon beta in context of LPS stimulation. Is this resulted then in a drastic reduction of IL-18 transcription, and that also worked in a concentration dependent manner. So the more type 1 interferon we block in these experiments, the less IL-18 expression we see, the black line. In contrast, it drives up the IL-1 transcription. And while we were working on this deeply in the experiments, this study kind of sent shockwaves through the people that have been working at this project. This was a study published by the Kaniganti lab in a journal of immunology back in 2017. and they reported data that phenocopied our human findings exclusively in mice cell. So this is IL18 and IL1 expression upon LPS challenge in mice, and look at the kinetics. You have the delayed expression kinetics for IL18, and you have the ad hoc kinetics for IL1. Exactly the same what we observed in humans. And then they do something which you can do in mice. In humans, we had to use other tools like blocking interferon signaling by antibodies or tofacitinib. In mice, you can use a type 1 interferon receptor knockout. And if you do that, you can see that the IL18 expression is basically brought down to baseline, whereas the IL1 expression is not affected at all. And you can also see that when you engage with other factors involved in the downstream signaling of type 1 interferons, all of these knockouts result in a blunting of the IL18 expression, whereas IL1 expression just does not care. So together, these human and murine observations point to a fundamental difference in the role of type 1 interferons in influencing IL1 versus IL18 gene expression. So type 1 interferons have a negative impact on IL18, on IL1 expression, but do positively regulate transcription of IL18. And with respect to IL1, that is actually something that has been described before. I mean, you know that, for instance, multiple sclerosis patients are being treated with type 1 interferons, interferon beta, to dampen inflammation. And the reason for this is actually coming from studies that look at the role of type 1 interferon and IL1 in bacterial infections and viral infections, because in case of a virus insult, what happens is that the immune response is very much biased towards expression of type 1 interferons. And what these type 1 interferons then do is they result in expression of genes that can impair IL1 signaling. So it's the expression of the IL1 receptor 2, this decoy receptor for IL1, its expression of IL1RA, the antagonist to IL1 signaling, as well as IL10 to cool down overall inflammation. In turn, if the host needs to combat a viral infection, IL1 signaling results in the expression of prostaglandins, which can then in turn reduce expression of type 1 interferon. So it's a balance of the immune system towards either combating a virus or combating a bacterial infection. And in fact, IL18 has been implicated since a while already in viral defense, and it's been known that type 1 interferons are required to recruit inflammatory monocytes to the site of infection, where they sit and produce or can produce, among other cytokines, of course, IL18, and IL18 is then signaling to the NK cells. NK cells produce interferon gamma, and that is central to control the viral infection. And viruses also knew how important IL18 is for the host to defend against being infected. So some viral variants, like pox viruses, have encode a protein that operates as IL18 binding protein. So these viruses can sequester IL18 and prevent and thus break this access of IL18 towards interferon gamma as an evasive strategy, which I think is extrememing fascinating. Now, in light of our data and the Kaneganti Labs data, redrawing this balance a little bit, means that we have type 1 interference as central regulator, actually, whether the system is focusing, the immune system is focusing on defending against a viral, a bacterial infection via IL1. So then IL1 is over prevalent. It's reducing type 1 interferon expression, which in turn can then less promote IL18. On the other hand side, if the immune system is focusing on viral defense, we have a lot of type 1 interferons around that can then push IL18 expression and in turn cool down IL1 expression. So type 1 interferons are really central in helping the immune response or shaping the immune response to either viral or bacterial defense. So following this, and now I'm taking a shortcut, not going to guide you through all these data, we asked now that we've seen the prominent role of type 1 interferons in all of this, can they induce IL18 expression by themselves? And in short, the answer is no. Instead, the model that we developed at that point is that it requires an inflammatory signal coming in via TLR that results in inflammatory cytokine as well as type 1 interferon expression. And then the type 1 interferon here comes together with the inflammatory signal, needs to synergize with the inflammatory signal to result in transcription of IL18. If there is excess type 1 interferon around, we have excess of IL18 transcription and corresponding cytokine production. And while we're putting together the paper, we found something in the literature, a real treasure in the literature that again phenocopied our data in this respect. And what these people did, they looked at IL-12, P70, and IL-18 on the side. They weren't in fact so focused on IL-18. They just dragged it along in their experiments. And they treated mice with LPS, single shot. See, they got IL-12 expression, some IL-18 expression, as well as a double shot. So it's actually, again, this endotoxin re-challenge model in a one or a double challenge with LPS, like I showed you in the very beginning of this lecture. And what you can see on the level of IL-12 is, again, immunoparalysis, double exposure to LPS, blunts the IL-12 expression. Instead, it fires up the IL-18 expression, exactly like we have seen it, both in humans in vivo as well as in vitro, upon double challenge with LPS. And then they did something in addition. They combined a LPS with CpG, which is a professional type 1 interferon inducer. And you can see some elevation on the side of IL-12, but you can see significant and strong elevation on the side of IL-18. So the combination of an inflammatory plus a type 1 interferon inducing trigger is pushing the IL-18 expression to the max. And going back to the biological relevance in course of viral infection concept, that puts an additional layer on this, saying that it's not just type 1 interferons that push IL-18 expression here, responsible for IL-18 expression. Instead, what we really need for IL-18 production is an additional inflammatory environment, which can already be eminent in the host before, but can also be tissue damage or superinfection. So additional inflammatory triggers need to come together with type 1 interferon to facilitate IL-18 expression in course of a viral insult. Now, what does that have to do with macrophage activation syndrome? Because that's what we want to talk about in the end. And the model I want you to look at is something that has been generated by Alexi Gram and Anna-Karin Horn and Fabrizio and Benedetti some years ago. And whenever I show this slide like this, Fabrizio complains that I'm cutting out the genetic contribution here, but I think it suffices that way. So according to this model, we have background inflammatory activity. like we have it in systemic JA, for instance, due to inflammatory cytokines and DAMs. And then we need an additional trigger on top, which can be any kind of infection, for instance, or other triggers we have not determined yet. And that then sets off the cytokine storm and macrophage activation syndrome. And with respect to trigger, this particular work states, and I'm quoting, that episodes of MAS in systemic JA commonly occur when elicited by viral infections, because they're known to activate interferon gamma-induced pathways. And if we recall this Annals of Rheumatic Disease case report from 1985, they also already suspected a viral insult in combination with a cytokine storm, hyperferritinemic cytokine storm they observed in that patient back then. So there is some clinical evidence that viral infections are associated with onset of MAS to some extent. And in fact, the Murine models, which we use these days for mimicking these conditions in an experimental setting, exploit exactly inflammatory trigger, type 1 interferon triggers, so triggers that simulate viral infections. So the prime model has been reported by Ed Behrens and is based on a repeated challenge of mice with CpG, which is a ligand to toll-like receptor 9. And that model can be run with or without blockade of the IL-10 receptor to give a more moderate or a fulminant phenotype that is resembling cytokine storm and macrophage activation syndrome. And lately, there has been a second model being reported, and that model already acknowledges the combination of an inflammatory trigger, LPS here, as well as a viral insult, or viral insult simulating trigger, which is poly-IC. And what they report is upon combination of these triggers in a specific manner, so poly-IC first, followed by LPS, this has a tremendous impact on the survival of the animals that have been challenged that way. And it overall gives features in these animals that resemble MAS and cytokine storm syndromes. So again, here, combination of type 1 interferon induction plus inflammation is giving severe hyperinflammation. So based on this concept, now these models, our proposed model looks like this. In systemic JA, we have background inflammation, we have eventual infectious triggers not identified yet. We certainly have damage-associated proteins around, like S100 proteins, very high in systemic JA, which can hit TLR4, but also other receptors that send inflammatory signals inside immune cells that then express inflammatory cytokines. Type 1 interferons are being produced either upon TLR4 expression or by other ways, hit the interferon receptor, and then these signals synergize to produce IL18. Now, if there's a viral insult on top, you have a professional type 1 interferon trigger. So you have a lot of type 1 interferon coming into the system that is already on a constant baseline inflammatory activation. And these signals then together, inflammation and type 1 interferon, generate IL18 expression, IL18 production, which can then, via interferon gamma, result in MAS manifestation. Now, assuming this, we thought of a potential therapeutic relevance. And this course is about treating autoinflammation. Now, let's get to that. And the therapeutic relevance we're assuming here is that a JAK-STAT inhibition might be able to break the IL-18 interferon gamma axis already upstream of IL-18 by neutralizing the type 1 interferon signaling here. And we have seen in the in vitro data I've already showed you that tofacitinib treatment of cells had exactly that effect, but that was in vitro. We wanted to look into this in vivo, and therefore we teamed up with a group of Kim Nichols at St. Jude's in Memphis, and Sabrine Albaitouni was doing these great experiments. And Kim's group has a long-standing experience in running the mouse model upon using repeated CPG challenge combined with the IL-10 receptor blockade. And in particular, she has experience in treating these mice with the JAK-STAT inhibitor, which is ruxolitinib here. And they have already done that back in 2016, reporting the power of a JAK-STAT inhibition and lessening inflammation in this particular model. And they've followed up on this in a mechanistic manner, also involving interferon gamma blockade here. And this is just an exemplary plot from this paper showing that on the level of body weight of the animals in this model system, is that ruxolitinib is the one drug here, the blue lines, that can really normalize the body weight. So the load of inflammation in this model, and it can do that more powerful compared to interferon gamma neutralization, and of course to the untreated animals, which is the yellow line here. So we relied on Kim's and Sabrine's extensive experience in this respect, and we were interested in looking at IL-18 expression in this particular model. And what we could see is that in both the moderate model as well as the fulminant mass secondary HLH model, the treatment of animals with ruxolitinib resulted in a lowering of IL-18 levels in serum almost down to baseline compared to untreated animals, which are always in the middle. So that was showing on in vivo level that an engagement with JAK-STAT signaling has a very powerful impact on IL-18 production. And we also had the chance to look at this in a patient together with Grant Schuler and actually in Cincinnati. They were seeing a patient with treatment refractory systemic GIA and recurrent episodes of MAS that was not responding to any treatment. And eventually at some point, they decided to use tofacitinib to get a grip of his overall inflammatory presentation. And you can see that upon initiation of tofacitinib in this patient, ferritin levels, CRP and other parameters, CXCL9, importantly, as I said, biomarker for interferon gamma signaling, are cooling down, coming down to baseline. And for the first time in that patient, never happened with any treatment approach before, it actually lowered the serum IL-18 levels as well. So we collectively, what we could see is that upon JAK-STAT inhibition in MAS, we can actually control IL-18, gain control over IL-18 expression, and eventually break this axis by interfering with the type 1 interferon signaling here. And in fact, the JAK-STAT inhibition has a double benefit because it not only targets the type 1 interferon signaling, it can also interfere with the interferon gamma signaling. So it's kind of a double hit therapeutic, eventual therapeutic strategy in controlling macrophage activation syndrome. And there's one other drug, these will be the very final slides, that I want to mention in this respect, and that is colchicine. So colchicine is been used by us, and you probably most likely as well, in treatment in FMF. And as I said, FMF is also a disease that is hallmarked by extremely high IL-18 levels. So what colchicine does is it acts microtubule depolymerizing. It's belonging to a class of drugs that is also termed microtubule depolymerizing agents, or MDAs. And what these drugs do, and that is something that has already been observed in the 1980s, destabilizing the microtubule skeleton has an impact on type 1 interferon expression. And that has been something that's been picked up again in the Nature Communications paper back in 2014. And this is where this graph comes from, illustrating type 1 interferon secretion by cells treated not or treated with colchicine. And you can see that this is really bringing down the type 1 interferon expression here. And this is something exactly in the same manner as we also could see in our experiments. If you treat cells in the LPS stimulation context with colchicine and no cortisol, both are microtubule depolymerizing agents, and we analyze the interferon expression over time, you can see that both on transcription as well as on interferon release level, treatment with these drugs is actually reducing type 1 interferon expression. And we were, of course, interested in what's happening to IL-18 if we do that. We could see that treatment of cells with colchicine or no cortisol resulted in a blunting of IL-18 expression. And this could be rescued completely when we on top treat the cells with type 1 interferon. So summing this up, we could see that this is again the model of double hit for IL-18 expression that is required. LPS and type 1 interferon need to come together to switch on IL-18 transcription. If we interfere with the cytoskeleton integrity using colchicine or no cortisol, that blunts the type 1 interferon expression and shuts off IL-18 transcription, and that can be rescued by addition of type 1 interferon here. So collectively, there are now different strategies to target this IL-18 interferon gamma axis in systemic JA mass. We can interfere with IL-18 itself using recombinant IL-18 binding protein. We can interfere with interferon gamma signaling using like emapalumab, the monoclonal targeting interferon gamma. But we can also interfere upstream of IL-18 with type 1 interferon signaling, which is using YAG-STAT inhibition, for instance, or treatment with colchicine. And that was my last slide. And I just need to acknowledge the many people that have been involved in all these studies, which is first of all my PhD student, now postdoc with Grant Schulet in Cincinnati, Emily, who did most of the hands-on experiments, and the many other people in our lab actually supported these studies. I acknowledge Peter and Alexia and Grant already in Nijmegen and Cincinnati, as well as Sabrine and Kim in Memphis, and also Claudia and Fabrizio have been involved in all biomarker studies heavily, and in ongoing collaborations, as well as our working party, as well as the funders. And I all invite you to Rome, in fact, in March this year. So very soon, there is a hybrid course on systemic JA and MAS that is organized by Claudia, Francesca, Bas, and myself as the core team of the respective working party of PRESS. We have a fabulous board of speakers that will touch upon all aspects that are relevant right now and state of the art right now in SJA mass passive physiology. So we will have a focus on lung disease, for sure associated with mass or generating emergency situation with mass, touch upon treatment strategies. We have patient representatives. So I all invite you to look at the respective website. You can register for online presence or on-site presence, even if possible. you can still submit an abstract with an interesting clinical case. So I would certainly, and we all would certainly look forward to see you there, and would love to welcome you to this course. And with this, I close and stop sharing. And I'm happy to discuss.
Thank you very much, Dr. Kessel. It's really interesting. It's a brilliant lecture. About the question, Dr. Mariana Marcos, thanks a lot. Dr. Kessel, I have a question. Have you seen any clinical evidence of using colchicine to treat patients with refractory MAS?
Very good question, very relevant. No, we have not. In fact, I mean, there are some anecdotal reports of people that use colchicine to control severe inflammatory episodes in systemic JIA, and it's working. But we in our clinic, in our outpatient clinic, have discussed that. In fact, every now and then, in course of when there are refractory patients that we or where we fail to identify a treatment that is really working consistently in these patients, and we can see them moving closer to MAS every now and then, and kind of generating real pressure for immediate action, then colchicine is sort of on the list every now and then. So I think there is nothing that really argues against that, from my knowledge, to use that. I mean, particularly in these cases that you cannot control otherwise. But we don't do it in a systematic manner, nor do I know of anyone else to do it in a systematic manner in systemic JIA.
Yes, thank you. And my question, what is your experience about the use of JAK inhibitor in refractory MAS? Did you use it in treatment?
Did you use it again? Sorry.
In a treatment schedule.
Yeah. So we use that already for short-term use, just to control severe inflammation. And I also know, I mean, there are some case reports around to date that did the same thing, successful usually. I mean, JAK inhibitors right now, you know that, have a bit of a problem. They're kind of flagged for generating severe side effects, and respective studies are paused or discontinued. And there has been a warning, I mean, sent out by the New England Journal not to use JAK inhibitors in certain conditions. But I think this is mostly due to a permanent use of JAK inhibitors to control chronic inflammation. And in my view, like in SLE, for instance, and in my view, it's not so overly surprising that this can cause problems, because in contrast to the very specific cytokine inhibitors, you of course hit different pathways. And it is impossible to tailor a JAK inhibitor, I think, in that manner that will only interfere with this or that pathway. I think this is a problem. However, the short-term use to control a hyperinflammatory condition is something that should still be on the menu for the use of JAK inhibitors, because in case of controlling MAS or also secondary HLH, it's not that we're talking about a use of these drugs for not even for weeks, probably. It's just a matter of days, likely, just to cool down the hyperinflammation, to break the overt IL18 and interferon gamma signaling, and help the system to normalize. and then to wean off and taper the JAK inhibitor and move on to another holding therapy?
Yes, in which step you suggest this? Before this, you suggested in first step, before the cytokine storm and inflammation, it's we should predict this situation as early as possible to start JAK inhibitor, yes?
Perfect question. I mean, this is the question right now. And in fact, I've just, with other colleagues in Europe, we just put together a grant proposal that is addressing exactly this question, because the situation right now is that we have very good biomarkers to assess, I mean, as I said, support the differential diagnosis of macrophage activation syndrome, as well as we have markers to assess the activity of the hyperinflammation in patients. We have all of this. What we do not have, or what we can't do with these markers to date, is predict when a patient is moving closer to macrophage activation syndrome, or the holy grail maybe, to predict early on, really early on, which patients are more prone to fall into macrophage activation syndrome and who don't. Because you know that you can have a patient that is perfectly stable on his or her medication or not, and both of them have the same risk to develop MAS. And it would be perfect to predict that. Right now, we can't do it. We try, though. So we have a collaborative study ongoing with Fabrizio's group in Rome, where we are looking at type 1 interferon score to understand whether we can see sort of an elevation at type 1 interferon early on before the onset of macrophage activation syndrome. And the hope is, now coming to your question, when do you treat? The hope is by identifying signatures and biomarkers that would have the power to predict MAS, to really open an adequate window of opportunity to come in with a JAK inhibitor, for instance, because I would think the earlier you see MAS to develop and the earlier you can come in with a JAK inhibitor, the more likely it is to be effective. That's what I think. But we need to define this window, and we need to have tools to really capture these patients really early. So I'm totally with you there. It's an excellent question. And I think the burning question right now in the whole MAS biomarker complex.
Yes. And the hot topics today, COVID-19 pandemic. We are rheumatologists, we are involved in this situation when they say that it's a hyperinflammatory syndrome, such as macrophage activating syndrome or cytokine storm, 1 to 2 months after COVID-19 infection. In adults, they say that not in mild and moderate hyperinflammatory syndrome, but in severe hyperinflammatory syndrome, we have the decreasing level of interferon 1. But in pediatric patient, after COVID-19 infection, the hyperinflammatory syndrome after COVID-19 infection, like Kawasaki disease-like syndrome, that they say like this, or MISC, multisystem inflammatory syndrome in children. They say that we have the increasing level of interferon 1 with sting stimulation. The stimulator of the interferon 1 gene expression in this children is against of adult patient. But both of them, we have the higher level of, for example, interleukin 6. And as mentioned, I read your nice manuscript about the discrimination of COVID-19 hyperinflammatory syndrome from macrophage activating syndrome in pathophysiology. Would you please explain more about this? Thank you.
Yeah, sure. Well, when the the thinking or the theory emerged that in context of COVID, or a severe COVID-19 would involve cytokine storm conditions. There was, I mean, this discussion in the beginning was very much, if not exclusively, hinged to IL-6. I mean, there's so many reports coming out that people said, yeah, IL-6 is high in these conditions, so it has to be cytokine storm. And this is the point where we thought, well, hang on for a second. I mean, IL-6 is certainly an important inflammatory cytokine, but I mean, we as rheumatologists or scientists that investigate these conditions know that IL-6, yes, it is elevated and it is high and it is important, but it's not the cytokine that is driving this cytokine storm and relevant for this. And that's why we set out and said, I mean, we need to compare to be really sure what we're talking about. We need to take defined classical cytokine storm syndromes and compare that to the situation as we have it in severe COVID-19. And in fact, this study highlights, well, it shows IL-6 is certainly high in COVID, absolutely. But what you can also see is that classical cytokine storm syndromes are really dominated by this IL-18 interferon gamma axis, and we do not see that to the same extent in COVID. And when we saw these data, we were actually kind of disappointed in the beginning because we thought, we just had this really nice mechanism, we thought, with the type 1 interferons that would actually push the IL-18 expression. But we didn't see it in COVID. And we said, aha, that's really interesting. It was kind of a bit of a downer. But I mean, only shortly after we published our data, there was the report that illustrating that in severe COVID-19, there is indeed a problem with the type 1 interferon expression mediated by anti-interferon type 1 interferon antibodies, as described by Paul Bastard in the Casanova lab. And that kind of sort of put, for us at least, things in the right perspective, again saying, okay, type 1 interferon expression is lowered in COVID-19, and that would then associate with also generally lowered IL-18. So yes, I mean, there is lower type 1 interferon in adults in COVID-19, associated with general lower IL-18 and interferon gamma expression and impaired viral defense that way. And that is certainly different from the classical cytokine storm syndromes as we have talked about now in the last hour. In MIS-C, the situation is different. I mean, there we actually have a really prominent activation of the innate immune system. Here, immune cells do not seem compromised, at least in type 1 interference and the respective defense mechanism that those trigger. And therefore, MIS-C, I think, is really different, and all the data show that, is different from COVID-19 in adults. And I think just yesterday there was a multi-omics paper published in Nature Medicine comparing the immune features in severe COVID-19 with MIS-C. So MIS-C is closer also from our data, which we are generated actually right now, much closer to the cytokine storm syndrome, the classic cytokine storm syndromes, than is COVID-19 in adults.
Yes, yes, thank you. And about the interleukin 1 in the cytokine storm macrophage activating syndrome. In one of your slides, you discriminate these two phenomena, MAS from secondary HLH, I think. And I think, what is the location of interleukin 1, anti-interleukin 1? For example, IV anakinra.
Yeah, sure. In controlling inflammation, you mean the mass, yeah. No, I mean the use is, it's very effective. I mean, we know that, right? I mean, anakinra treatment in course of MAS is an absolute key therapeutic strategy and necessary, and it's working. in most cases. There are cases like the one patient I showed, and there are other refractory cases with recurrent episodes of MAS that do not respond to IL-1 for reasons that we do not understand. As there are systemic JA patients, we also see them. They have full-blown inflammation, full-blown features of disease. They do not respond to anti-IL-1. We don't understand why. Same situation as for predicting MAS. It's another sort of holy grail in this class of diseases. And we have studies ongoing on this to predict the patients that respond and do not respond to IL-1 inhibition.
Yeah.
But for controlling hyperinflammation, I think the use of IL-1 in course of ongoing hyperinflammation is, it's justified.
Yes, my another question in polygenic type 1 interferonopathies like SLE and GDM. But would you please explain more about MAS in this situation? I think JAK inhibitor would be helpful in this situation. Yes, is it okay?
Yeah, would be helpful. Again, I think, as I already said, I think short-term use of JAK inhibitors in controlling hyperinflammation, that's at least what I think would be the way to go. Disease mechanism of MAS and SLE is underexplored. I think what we know is in SLE, you certainly have constantly elevated type 1 interferon expression. And accompanied with that, there is a very good correlation with that. We have elevated IL18. So the likelihood that in SLE MAS, you have the very, very same axis operating, and the same is true for JDM, by the way. You have the very same axis operating as we have that in systemic JA MAS is really high. But I think it's probably the very same mechanism that drive the disease. Just the basal inflammatory setting is a bit different. I mean, in systemic JA, we have IL-1 signaling, and we have potentially DAM signaling that generate this basal inflammatory environment. In SLE, you have a different setting there. The basal inflammatory environment is more, or it's interferon dominated, type 1 interferon dominated. And then at some point, you probably have an inflammatory trigger, not a viral trigger. You don't need that in this scenario because you already have so much interferon. You have an inflammatory trigger that is then pushing or results in patients in falling into MAS.
Yes. And excuse me, I have a lot of questions.
Sure, go.
Yes, about in our country we have such patients such as secondary HLH in the background leishmanios disorder, the parasite disorder, the secondary HLH in parasite disorder, and which is in priority, the treatment of the parasite or a treatment of this cytokine storm due to this, because when we use the corticosteroid, for example, the patient go to the progress to the to the worst condition in some situation. What is your idea about?
This is a tough question. So which treatments do you use there to control disease?
Yeah.
Well, which treatments do you use to control? So what is your strategy? You're treating the leishmaniosis, or that is the primary treatment you use, probably, right?
Yes, yes, I think the treatment of parasite is in priority of cytokine storm, but we haven't enough time to manage this bad condition children. Parasite infection or the cytokine storm situation.
So which drugs do you have available to control cytokine storm?
Yes.
In your country, what do you have available?
I think
steroids?
Yes, steroids. We use the HLH 2004
criteria.
Yes, yes.
Yeah. The steroids in this respect are of course a problem, right? I mean, when you treat you come in with the steroids, that is not beneficial for the parasite infection. It's actually vice versa. So you may exacerbate one for the other. So I think this is a really tricky question to answer. I would think that in this particular scenario, an IL-1 inhibition would actually be better because it's more targeted than the steroids. But I don't know whether this, I mean, it's a parasite infection, and it's a bacterial infection. So IL-1 inhibition could also facilitate this to break through more. I mean, yeah, to be honest, I'm struggling obviously with giving like a clear management presentation, because it's a very special scenario, I think.
Thank you very much.
I'm sorry.
No, thank you very much. We are honored to have you here and want to use you a lot. Yes. About our colleague's question about Dr. Mariana says, do you think that CXCL9 might be a helpful biomarker to evaluate risk of MAS? I think about the JAK inhibitor uses in treatment in the first step.
Yeah. CXCL9 is an excellent biomarker, actually. It's evaluating the risk of MAS, then it depends on when you start. I mean, it's not useful in really early on, like prediction of MAS. CXCL9 is not of help. But as soon as a patient is moving towards MAS and also moving into MAS, CXCL9 is excellent to monitor the overall disease activity. And also, if you then initiate a treatment in such a patient, steroids or anti-IL1 or JAK inhibitors, CXCL9 is optimal it's a very good biomarker to monitor the efficacy of treatment. I mean, it's the primary biomarker used in the emapalumab study to understand the penetrance of this medication. So yeah, I fully recommend CXCL9. People usually ask whether they should also measure interferon gamma. Interferon gamma is really tricky to measure. It's mostly sitting in the tissue, actually, not even in the bloodstream. So CXCL9 expression is happening to a large extent in tissue. There it drives CXCL9 expression, and CXCL9 then moves to the blood where we can measure it. So it's a perfect surrogate marker of overt interferon gamma signaling.
Yes, thank you. And about the use of colchicine in treatment guideline HLH 2004 in combination with cyclosporine, is it good?
I think, I mean, looking at the data that there are, I think it's, I don't see something that would argue against it. Yeah.
Yes. And the other question about interleukin-1, and that we asked this, and about the tocilizumab. Can tocilizumab be administered as inhaler to local effect in COVID? Affected patient in hyperinflammatory syndrome, and for prevention of systemic exposure.
Inhaled tocilizumab, is that the question? As to support treatment in COVID? Did I understand?
Yes, I think we don't have this. Yes, we have.
I don't. No, I'm not aware of it. No, I'm just thinking. I mean, if possible, you would restrict the treatment to the lungs and respiratory tissue with this, I think. And just thinking that I mean, I think the IL-6 inhibition in COVID is, if it works, it's probably more on systemic level than just a lung restricted level. I mean, generally, the tocilizumab data in COVID are so-so, I think. Yeah, so it's a bit of a hypothetical thinking. Yeah.
And Dr. Dedoglu, we are honored to have you here, Dr. Dedoglu. She said that if Dr. Dedoglu, if you want to speak, we are honored to have your voice. Dr. Dedoglu, please raise your hand if you.
I am here.
Hello, Dr. Dedoglu.
So, hi, it was excellent. I woke up at 6 a.m. and, you know, I'm trying to still wake up, but excellent talk. So thank you.
Thank you.
No, actually, it's interesting that you mentioned colchicine because I'm like a desperate, and I'm about to start a patient of mine with SystemicJIA and MAS, and already on max on Ruxo and Cyclo and hypertension issues, max on steroids for a flare. So I was thinking, you know, to do the colchicine, so I probably will.
Yeah, I said, I mean, it's every now and then it's also with us on the list of handling difficult patients. And I mean, if there is a patient that does not respond to anything that you would otherwise consider, I think there is no, why not? Yeah, why not? And the data, mechanistic data, would be in favor of this.
Yeah, I actually, with systemics, I generally use like methotrexate or T-cell blockers like systemics that have a tendency to MAS and over all these years, and I've seen really good responses and so on. So I think like addressing T-cells with patients who have a tendency to MAS in systemic is really important.
Yeah, absolutely, totally agree. Yeah.
Thank you very much, Dr. Dedoglu. We are honored to have you here. Thank you. And thank you very much, Dr. Kessel. It's the brilliant session. Really, it's a brilliant session. We learn a lot from you, and
my pleasure.
We hope that we'll cooperate with you in future. Thank you very much.
Thank you. Thanks for having me. I really enjoyed this great discussion, and yeah. Let's collaborate and repeat this.
Great. I'm very thankful to you, Dr. Kessel, for a fantastic speech. It was very interesting and very informative. And I specifically enjoyed the way you presented this much of invaluable data. Thank you very much.
Thank you.
Thank you. And we are honored to have had this experience for having this session with you. And we're looking forward to the next collaboration for sure.
Great. Thank you.
Thank you very much. And I would also. You too. Bye. And I would also like to thank Dr. Asari for being the great moderator of this fabulous session. I am grateful for participation of all you academics in this session of How I Treat Autoinflammatory Disorders course. I hope you have had an enjoyable time. We will share the posters later with the participants, and then we can send the certificates. The first 3 session certificates have been distributed up to now. As I mentioned before, the course certificates are approved and validated by OPLAR and Pediatric Rheumatology Society of Iran, Iranian Rheumatology Association, and Tehran University of Medical Sciences. We invite you to our next session, which is going to be held next week, February 25th, entitled as Interferonopathies with a special aspect on rare portal gemopathies. And our speaker is Dr. Elke Kruger from Germany. You can find the details on the next sessions in our website, and you can see the link in the chat forum. Also, we will be very grateful if you can fill in the feedback form that we shared in the chat forum. Thank you very much and have a great day.
Thank you. Bye-bye.
Thank you. Goodbye. Bye.
Thank you. Bye.